At the top of the priority list for the Preventive Technologies Agreement was addressing women's risk for HIV infection. Women, especially women in sub-Saharan Africa, have particular HIV risks and often are unable to protect themselves from HIV infection.
These risks are related to gender, economic, and educational disparities. Current HIV prevention methods largely fall outside of women's control. For this reason, 2 clinical trials of HIV prevention methods controlled by women were at the heart of the Preventive Technologies Agreement. The first trial, Caprisa 004, assessed a vaginal gel containing the microbicide tenofovir. Caprisa, the Center for AIDS Prevention Research in South Africa, led this trial. This country ownership was a global first.
With support from USAID, FHI 360 was really very proud to be part of the Caprisa 004 clinical trial, which was the world's first study that showed that a woman-controlled vaginal microbicide could prevent HIV infection, and it was the first study that showed an antiretroviral agent could prevent HIV infection. So FHI 360 partnered with Caprisa and local communities in South Africa, enrolled a number of very high-risk women, and showed that this product, if used consistently, could reduce a woman's HIV infection risk by about 40%.
And secondary analyses showed that if women adhered to the regimen for using the gel, that the effectiveness was higher. So in the aftermath of Caprisa 004, there are several questions that we would like to see addressed, and that would be both FHI 360 and Caprisa. We want to know if we roll out tenofovir gel in a family planning context, what's the impact for health service delivery? If women are receiving tenofovir gel in a family planning context, can they still adhere to the product? We know that some women can disclose tenofovir gel use to partners, but others are challenged. We want to know what we can do to support women who are challenged in disclosing tenofovir gel use to their partners. We're working with Caprisa to implement a study looking at these social, behavioral, and health service delivery issues. We're expecting to have results available by the end of 2014.
Under the Preventive Technologies Agreement, FHI 360 also led the development, implementation, and analysis of a clinical trial called FEM-PrEP. It tested whether a pill called Truvada could prevent HIV infection among high-risk women. In April of 2011, we had an interim meeting of a scientific review committee, and they determined that the trial would not be able to conclude that Truvada was effective for HIV prevention. Secondary analyses that we did thereafter showed us that women did not take Truvada often enough in the trial for us to be able to demonstrate that it's effective for HIV prevention. So in FEM-PrEP, we learned that adherence is absolutely critical in both evaluating a product to determine if it's effective and in rolling out a product in the future to actually help people.
I think FEM-PrEP was a successful trial because we were able to gather very insightful and useful data on the reasons why women join clinical trials. We were also able to learn reasons why participants choose to take an investigational product in the clinical trials and reasons why they don't take an investigational product during these trials. And these data can be highly informative of future HIV prevention clinical trials. One factor we found to be associated with higher adherence was risk perceptions. So women who had some perceived risk of acquiring HIV were more likely to take the study drug. But what we also found is many of the women who seroconverted perceived that they had no risk of becoming HIV infected. At the end of the day, women continue to be at substantial HIV risk in sub-Saharan Africa, and we need new HIV prevention products. But in order to get women to use these products more often, we need to continue our research to better understand risk perceptions and risk rationalizations among women.
So the preventive technology really made a very substantial contribution to the world's effort to prevent HIV infection, especially for women. The Caprisa 004 study for the first time showed that an antiretroviral agent in the form of a vaginal gel could effectively prevent HIV infection in very high-risk women in South Africa. And the FEM-PrEP study showed that oral drugs have to be taken very regularly to be effective. Both of these findings really allowed us to have a better understanding of women's perception of risk, their ability to use a product, and have led us to improved ways of administering drugs to women at high risk for HIV infection.